Protein synthesis. Code for protein synthesis is specified by DNA and has to be sent to ribosome. From where the protein synthesis takes place is it begins from the DNA code. So generally, in a human body, proteins can come from two sources, from food and from the amino acids exist in the body. So this part is protein synthesis. DNA is a huge molecule and remains in nucleus to assemble amino acids in the correct sequence to form protein. Events during protein synthesis there are different stages or events. The first one is transcription. DNA code for protein is rewritten in a molecule of messenger RNA or mRNA. Transcription, trans means across and cription means writing. So the DNA code is just transforming into RNA code. That is it. The other event is mRNA travels from nucleus to ribosome. Where the mRNA made through the transcription process is in nucleus, because the genetic material found in the nucleus. Then it made in the nucleus, then transported to ribosome, because where the protein synthesis takes place is the ribosome. The other event is free amino acids are transported from cytoplasm to ribosome by transfer RNA or tRNA molecules. So mRNA moved to ribosome, then amino acids from cytoplasm will be moved to ribosome by tRNA. The other event is ribosome read mRNA code or the codon and assembles amino acids presented by tRNA. Presented means brought by tRNA or transfer RNA. Then this process we call translation. Translation is a process in which mRNA code is converted into a sequence of amino acids. Therefore, the sequence is DNA code, then mRNA codon, then protein or polypeptide. The final product is the protein, because the process is what? Protein synthesis or protein making. What is genetic code? The genetic code is held in the DNA molecule. As we said, the code ultimately found is in the DNA. That we call the genetic code. The code for protein synthesis. It is because of the nucleotides sequence in the DNA molecule made a gene that coded for protein to that each amino acid in the protein is coded for by a triplet or sequence of 3 bases. Tri means 3, so 3 bases will code for 1 amino acid. Therefore, a gene is a sequence of base triplets in the DNA molecule that carries a code for a protein. So we said gene is a section of DNA which will be expressed to protein. Since there are 4 bases, adenine, guanine, cytosine, and thymine in DNA, there is 4 cube, 4 to the power of 3, which is equal to 64 possible triplet codes, but only 20 amino acids are used to make all different proteins. There are 20 standard amino acids which will be made by this process, but there are 64 triplets. None of them is spare or redundant. From the 64, there is no redundant or there is no spare. That is idle triplets there. All will be in the squad and then play the role. Only one strand of a DNA molecule carries the code for proteins. It is called sense strand or coding strand. Unlike DNA replication, in the transcription process, only one strand of DNA is used as a template. That is, in DNA replication, the two strands are used as template. Okay, the two strands are used as a template. But in the transcription, only one strand used as a template. The other strand is non-coding, or we call antisense strand. Just template strand it means antisense strand. It is also called plus or positive strand. Then it is transcribed into mRNA. This strand will be transcribed into mRNA. The main difference between template and coding strand is that template strand only serves as the template for transcription, whereas coding strand contains the exact same sequence of nucleotides in the mRNA, except thymine. So, the coding strand is equal to mRNA, except thymine. Instead of thymine, then you will replace uracil. Therefore, the coding strand and mRNA will be the same. The template strand, you can consider what is template. Okay, the template is used to make the new from that. So the template strand is the strand which serves as the template for the mRNA synthesis during transcription. Usually RNA polymerase, which is the enzyme involved in the transcription of genes into mRNAs, adds nucleotides in the 5' to 3' direction to the growing strand of mRNA. Therefore, the template strand should be directed from 3' to 5' in order to add complementary nucleotide to the growing mRNA strand in the 5' to 3' direction. This is very interesting concept in DNA replication, in the protein synthesis. Here, this is a double-stranded DNA, because DNA is double-stranded. Here is the red colored from 5' to 3' to this direction, and this one is the green colored from 5' to 3' from this to this direction. And there will be what? Unwinding by helicase. Then consider this 3' will be the template. Okay, the green one is from 3' to 5' to this, so it is a template. When you go back to the previous statement, you will see that. So the template strand is 3' here, TACGCA, then continue. You can consider hundreds, thousands, millions of sequences here, depending on the protein will be made from the process. These are the stop sequences. Okay, ACT, ATC, ATT. These are one of the three. If there will be one of the three sequence encountered, then the process will stop. So from this, there will be what? mRNA, messenger RNA codon from the template. Then T with A with T, but it is RNA molecule, there is no T, therefore replace T by U. So U, then C with G, GC, CG, AU. For this, A with U, C with G, T with A, then A with U, T with A, and then C with G, then A with U, T with A, T with A. So these are, we call the stop codons, UGA, UAG, UAA, as a messenger RNA. As a template, these are the stop codes. At the coding DNA strand, there are TGA, TAG, and TAA. So these are the anticodon. Therefore, you can see anticodon, the template and the anticodon are the same. I made it in the green. They are 3' at this end, then UAC. In this case, TAC. Because this is a DNA, this anticodon is a transfer RNA, so it is RNA. There is no thiamine. Instead of thiamine, you will replace by U, so UAC, except that they are the same. And the coding DNA red and the messenger RNA red, they are the same, except U and T, the variation of U and T. Just if you can understand these things, just you have understood everything in the genetics. Very interesting. The other event is most amino acids have more than one code. Only methionine and tryptophan have one code. For example, arginine has 6 codes. 3 triplets, these are TAA, TAG, and TGA. Considering which strand from the previous TGA, this is coding DNA strand. Coding DNA strand. Considering the template strand, ACT, ATC, and ATT are the complementary triplets for the stopping codons. These do not code for amino acids and they are called stop codons. Stop codons signify the end of the coding sequence. Then when this one of the 3 codons encounter, then the process will stop. Degenerate code, more than one codon encodes for the same amino acid. For example, AAA, AAG is for amino acid lysine. We call this kind of feature degenerate code. The genetic code is also universal code. That is, which codes methionine in human does the same in prokaryotes. Okay, they are the same. The code AUG, the codon AUG for human is also AUG in prokaryotes, that is bacteria. This point is not exactly true as recently. That is because scientists have discovered many exceptions from this rule. The genetic code is not universal but still predominant. So science is dynamic and scientists are always in the work. So what you know yesterday may be modified today or tomorrow. This is a genetic code. Okay, you may find in the tabular form, but this is in the circular form. To read this genetic code, it starts from the central circle. There are 3 circles: 1, 2, and 3. For example, AAA lysine, AAG lysine, and the stop codons here is, for example, UGA stop, UAG stop, UAA stop. So these are the stop codons. You can read in such a way. For example, here GAC is for aspartate, and GAU also for the same amino acid. GGC glycine, GGU glycine. Okay. This is what we call the degenerate feature. You can see from 5' to 3' in this direction. For example, let's read for the first amino acid in the protein synthesis. Always the first amino acid is methionine, AUG. Where is A is here, U, G here. AUG, methionine, that is start codon. Says start. Notably, it is not just the base that matter, but the sequence. Okay? For example, ATT codes for different amino acid from TTA. The bases are there. A is there, here and here, T, 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 T. But the order matters, the sequence matters. Therefore, they code for different amino acids. The enzyme DNA-dependent RNA polymerase. This is the enzyme RNA polymerase, which is dependent on the DNA. Binds with a section of DNA next to the gene to be transcribed. Okay, during the transcription process, what will be made first is this process. RNA polymerase will be bound. Transcription factors activate the enzyme. Which enzyme? RNA polymerase. There are transcription factors. These are proteins which activate this enzyme. The enzymes then unwind and separate the strands of a DNA molecule. That is, helicase will unbind the helical structure of the DNA. As the polymerase assembles, that is DNA polymerase. This is an enzyme. This assembles free RNA nucleotides on the complementary strand, that is antisense strand. Then mRNA, then that is transcribed from the transcription process. It leaves the DNA, the strands of DNA rejoin and recoil. After the process of mRNA making, then mRNA will leave the template strand, then the old DNA strands will rejoin together. Again, note that the mRNA molecule now contains the code for the protein that was held in the DNA of the genes. That is it. The DNA, the code for a protein found in the DNA is converted into the mRNA. Translation of mRNA code into a protein depends on the interaction within a ribosome between mRNA and tRNA. Okay, there will be interaction between these two RNA molecules on the ribosome. All tRNA molecules have same basic structure. The clover leaf configuration of the molecules has at one end a triplet of bases called anticodon. The anticodon is a triplet on tRNA. The codon on mRNA, the anticodon on tRNA. We will see the structure of tRNA. The other end of tRNA molecule has an attachment site. For attachment site is what? The site for the amino acid to be attached. That is specified by the mRNA codon. Which kind of amino acid will be attached on the attachment site of the transfer RNA? It depends on the sequence of triplets on the mRNA. Ribosomes are made from ribosomal RNA and proteins organized into a large and small subunits. A ribosome is made up of ribosomal RNA and proteins. And then there are two subunits, larger ribosomal unit and smaller units. Within ribosome, there are three sites called A site, P site, and E site. What is P? For peptidyl. Peptidyl related with the protein bond. The second binding site for tRNA in the ribosome, P site. A site, A for amino acid, the first binding site in the ribosome, and the E site, E for exit, the third site. This is a structure of tRNA. Here is a amino acid attachment site at this end. Okay, for example, this is a methionine, one of the amino acids, the 20 amino acids. At this end, it is anticodon, UAC. This is for the complementary triplet for the AUG, which is codon for the amino acid methionine. Because A always with U, okay, so AUG, again U with A and then C with G. So here is the process amino acid attachment. For example, here is phenylalanine, an amino acid, as the amino acid attachment site of the transfer RNA as a 3' end here. Then here is the anticodon on the transfer RNA, then codon on the messenger RNA. This is a messenger RNA. Here, the same process. This is a ribosome. This is a larger unit, subunit, and this one is a smaller subunit. Then here is a messenger RNA attached on the ribosome, and then here is the first amino acid. Then the process will continue. For example, methionine, then tryptophan, then it will make the bond, then the protein will made finally. The same structure, just the larger ribosome, the smaller ribosome, then messenger RNA, then tRNA brings an amino acid, then finally there will be the protein, growing protein chain. This is the growing protein chain. Here consider methionine. Here the first amino acid brought by the transfer RNA, then again another amino acid, then finally we will get the protein. Thanks for watching.